The overall goal of this procedure is to perform robust, non-targeted metabolite profiling of serum in a large-scale experiment. First, extract the metabolites in a plate format. Establish a robust QC sample and method to ensure analytical stability over time. Then acquire mass spectral data on QC and randomized serum samples in replicate. Proceed to data pre-processing, statistical analysis, and metabolite identification. The results of non-targeted metabolite profiling by UPLC-MS can be utilized to identify small molecule biomarkers in serum.
The protocol is presented here for the analysis of serum. However, it can easily be extended to the analysis of other biological fluids such as urine, tissue, and cell culture extracts.
To create a plate map for sample preparation, open a spreadsheet. In the first column, enter the sample list in order of loading. In the second column, enter the 96 well plate locations using correct nomenclature for your autosampler software. Save one well in each plate for the QC sample. If your LC autosampler can handle 2 plates at a time, separate the sample list into batches of 190 and save this information in a second worksheet. Within each of these batches, randomize the injection order. Now copy and paste your injection order into the LC-MS sample queue software system. Reserve the first 3 rows of the sample queue for column conditioning injections.